Good evening. Thanks for joining. We will begin in a moment. We want to give everyone a chance to jump on. We'll begin the webinar in just a few moments. Hello, everyone. Thanks for joining. All right, looks like we have several people on, so let's go ahead and get started. And we'll advance to the next slide, Justine. Hello, everyone. Thank you for joining us for tonight's webinar as we discuss understanding your genetic variant, what it means, and how to take action. My name is Nikki Butler, and I'm the Director of Digital and Social Media for the Dilated Cardiomyopathy Foundation and the Genetic Cardiomyopathy Awareness Consortium. We're so glad that you can be with us as we explore what genetic variants mean. We have over 270 registrants for this educational discussion. Next slide, please. A few housekeeping items. All attendee microphones are on mute upon joining so that you can hear our presenters. Please enter your questions into the Q&A feature of Zoom at any time. There will be a dedicated Q&A session following tonight's presentation. Next slide. We do have a poll for tonight. So Justine, let's push forward poll number 1. We'd love to know who's attending tonight's webinar. Are you best described as a cardiomyopathy patient, a family member or friend, a medical professional, or an industry professional? It's always interesting to see the breakdown of these results. And we had so many people interested in tonight's webinar topic. Take just a little more time there to get your answer in. Right. So it looks like we have about 80% cardiomyopathy patients, 11% family member or friend, 6% medical professionals, and 4% industry professionals. So let's keep rolling. I do want you to know that this webinar is being recorded and will be posted on our YouTube channel following tonight's discussion. As registrants, you will receive an email with a link to the recording. Our YouTube channel also houses a library of our previous webinar recordings, and using the auto translate feature, these videos are available in 54 different languages. Next slide, please. To begin, I'd like to give you a little background. The DCM Foundation, focused on dilated cardiomyopathy, created the Genetic Cardiomyopathy Awareness Consortium, or GCAC, in 2023. This effort began in 2022 when the DCM Foundation recognized a major gap. Genetics and cardiomyopathy wasn't being widely discussed, not by medical professionals and certainly not by patients. And we realized something important. You can't talk about genetic cardiomyopathy in silos. Many gene mutations cross diagnostic boundaries. For example, a single gene variant might cause dilated cardiomyopathy, DCM, in one person and arrhythmogenic cardiomyopathy, ACM, in another. Because of that overlap, we needed a broader, more inclusive approach. So GCAC was formed to raise awareness and provide education about genetics and cardiomyopathy across all types. And next slide, please. In just a few years, we've made meaningful progress. Awareness of genetics and cardiomyopathy has increased significantly, both in the medical community and among patients. When we launched GCAC, we started with 8 patient groups, and today we've grown to 11, and we're continuing to expand. This momentum shows there's a real need and a real interest in understanding the genetic side of cardiomyopathy. Next slide. The 11 patient groups that are participating in the Genetic Cardiomyopathy Awareness Consortium, along with the DCM Foundation, are Laminate Cardiac, the Danon Foundation, Mended Hearts, PLN, SADS Foundation, Women Heart, Let's Talk PPCM, Team Titan, Heart Charged, and the Sudden Cardiac Arrest Foundation. We want to thank each group for participating. It's incredible to have a coalition of 11 different groups working together to solve a real problem with patients. And next slide. We will bring patient advocate Christy Johnson out later to moderate our question and answer session. Our primary speaker tonight is Brian Schertz, MD, PhD, president of Connect My Variant, and associate professor of pathology, microbiology, and immunology and biomedical informatics at Vanderbilt University. Dr. Schertz serves as medical director of the Molecular Genetics Laboratories at Vanderbilt, where he supports a wide range of clinical genetic testing. His research focuses on improving genetic sequencing analysis and strengthening communication of genetic test results among laboratories, providers, patients, and families. He founded Connect My Variant to help individuals with the same pathogenic variants find one another so they can share experiences, identify common ancestry, and support family outreach. His professional goal is to see the end of preventable hereditary disease. Please join me in welcoming Dr. Brian Schertz. Bring Brian on.
Okay, thank you, Nikki.
Thank you.
Found the unmute button.
All right, very cool.
I appreciate that kind introduction, and let me see if I can turn my camera on as well. I'm having a hard time finding my camera. Here we go. I'm looking forward to talking to this group, and I want to thank the GCAC for this invitation. So I'm going to be talking about how we can prevent hereditary disease by talking with your family members. But before we get into that, first I'm going to talk about what your variant is and how you can find out what your specific variant is on your report and what that means. So first, Connect My Variants. Our mission is to end hereditary disease by bringing families together. And we're based on the principle that if somebody knows about their genetic risk, then they're more likely to be able to have the right diagnosis, know how to prevent disease, and know what to expect. And most people don't know about their hereditary disease unless somebody else tells them about it. And the most common person to tell them about it is a family member who already knows about that genetic diagnosis. And that's been proven in medical genetics to be the most cost-effective way to do genetic testing. And unfortunately, it doesn't happen nearly as often as it should. So Connect My Variant has several techniques and tools that can help people find more family members. And I'll talk about that later on. Let me see if I can. Okay. Oh, I think I advanced twice. So the first thing I want to mention is that if two people have why do you want to know what your variant is? Why do you want to know your specific variant? And when I talk about specific variant, I'm not talking about the gene like Titan or DSP. I'm talking about the specific mutation. The specific, it's like the address that you would find on a house that shows exactly where that mutation is in the genome. And the reason why is that if two people have the exact same mutation, there's a 90% chance that they share that mutation or they inherited that mutation from a common ancestor. And if they find that common ancestor, then that can help them know which siblings, which cousins, which aunts and uncles to talk to, and which people in their family can be at risk. And also, bringing those big family trees together can help people communicate with each other and understand better what the risk is in their family and what their specific variant means for their family. So these are the types of things that Connect My Variant does. We have family outreach navigators, so we can help somebody kind of walk through if there's a difficult person to talk to, how they can talk to that person, who they should talk to next. We also have a monthly newsletter that helps people see success stories. We have some workshops that we do. And then the coolest things we do, I think, are actually these very variant-specific family reunions. When we get 3 or 4 or 5 people with the same variant together, we want to bring them all into the same virtual room so they can talk to each other. We want to help them build pedigrees. And we have partnership with patient advocacy groups like this one, and also partnerships with genealogists at the BYU Center for Family History and Genealogy. So we can give free genealogy assistance for someone who, let's say, knows their variant came from their grandparents, but but knows that they don't know all of their cousins because there was not good communication in the family. Or perhaps somebody who knows they inherited their variant from their dad, but their dad was adopted. So they don't know which aunts, uncles, and cousins they should talk to because they don't know their biorelatives. And so we have a lot of tools that we can help people really expand their outreach and talk to family members that otherwise wouldn't know about their genetic risk. So Connect My Variant's different than a lot of other organizations. We're not a research program. We're a public health nonprofit that's focused on education. We're really only focused on family outreach, so we don't do individual treatment. We don't do genetic testing. We're not going to tell you what types of things are associated with your specific gene or what types of risks you have. You should really be talking to your doctor about treatment options, and you should be getting you should be getting information from some of these foundations that have resources for specific genes about the types of treatment that might be available and the cutting edge research that's going on for your specific genes. We're personal and longitudinal. We have individual volunteers who talk to people. It's not something where we use chatbots or apps to be able to help with communication. Our goal is to build community. We're not trying to refer people to clinicians. We're just trying to make these family communities grow and to help people talk to each other. And then we provide genealogy assistance, which is kind of a weird thing for a health education organization to be doing, but we found that it's really important for hereditary disease. So, how do you find and interpret your genetic variant on your genetic test result? So here is an example genetic test result that I've been sent, and you can see kind of what it might look like. And here you can see there's an LMNA variant. And it says it's heterozygous, it's pathogenic. And there's also a factor 2 variant, which we're not going to talk about because it's not associated with cardiomyopathy. And so these are the components you see. You see the gene name. That's usually what you talk about most. And then you see this thing, the C dot. This is the coding change in the gene. It's the DNA sequence, and it says that at position 922 of the gene, there should be a G, but it was changed to A. And then also there's this protein sequence here. This is the protein change. It's how the DNA gets translated to RNA, which gets translated to protein. And this is what the proteins changed. And so it says that 331st amino acid has been changed from arginine to a glutamine. Because it's from one amino acid to another amino acid, that's called a missense change. Here's another genetic test result so you can get the idea. This is set up a little bit differently, but has a lot of the same information. So here you see there's the DSP gene, and there's a C dot 250 C to T. So that's, we'll get to that. And then here's a P dot here, and there's a classification as pathogenic variant, and we'll talk about that more a little bit more. And then here's another HFE gene. This is this is not related to heart disease at all, and probably is not very important for this individual. So we're going to focus on this DSP gene. But you can see there's sometimes multiple variants. So here's the DSP gene. Here's the C dot, which is the coding change, which is how the DNA sequence is changed. Here's the P dot, which is how the proteins changed it. And this tear is something that's interesting. So, I should say 84th building block here. I copied and pasted 250 wrong. And this tear means that, or sometimes there's an asterisk there. That means that it's not translated. It means that there's a stop code on there. And so after that part, the protein isn't translated. So this is called a nonsense variant. So here's another one. I don't have the report for this one, but here's another type of variant. And here's the gene name. Here's the C dots that tells which which part of the gene is changed. And you can see this C dot has the two positions, 1477 and 1478, have been deleted. And then this shows that the protein change means that there's going to be a frame shift here. And so that means that there's three nucleotides at a time code for an amino acid. And so that frame shift means that it's kind of like reading along a sentence, and putting the spaces at the wrong place. If you put the spaces at the wrong place between the letters in the sentence, then the words don't make any sense. And that's what a frame shift does. Okay, so you saw on that report that there are some variants that were said heterozygous and pathogenic. And so some variants in different reports are pathogenic. Those are disease-causing variants. And sometimes you see variants that are benign. So those are not expected to cause disease. And actually, you probably don't see those on your genetic test report because they're usually left off genetic test reports. Whenever you get genetic sequencing done, you're going to see lots and lots of common variants that are benign, that are seen in the population all the time. And we all have them, but they're not going to make any difference for the genetic testing reports. So they usually get left off of those reports. Sometimes we'll see variants of uncertain significance. That means that the lab is unsure whether that genetic change causes a problem or not with the gene. They're not supposed to change medical management, and familial testing is not generally recommended for variants of uncertain significance. And that being said, if there's multiple people in the family who have a variant of uncertain significance, then sometimes that information can help classify the variant of uncertain significance as benign or as pathogenic. Overall, about 80% of variants of uncertain significance end up being classified as likely benign. In some genes, that's higher. Like in Titan, then over 90% of variants of uncertain significance end up being classified as likely benign. Okay, so inherited disease looks different than non-inherited cardiomyopathy. Usually it happens at a younger age. Oftentimes you'll see sudden death at a young age. Sometimes you'll see cardiomyopathy with arrhythmia. Sometimes you'll see muscle fatigue. Sometimes you'll see ECG changes that you wouldn't expect with other types of cardiomyopathy that give hints that something might be genetic. But well, and the other thing is when there's multiple people on the same side of the family that have the same constellation of syndromes, uh, then that can also the same constellation of disease that can also help point towards something that's familial. But many families with hereditary disease don't have any of these features. You may be thinking about your family and thinking, that's not what happened in my family at all. And then there's also sometimes you see early onset disease where you see some of these changes and you do genetic testing and there's no hereditary disease risk at all. And so these are general things that make hereditary disease stand out, but they're not uniformly present. That's why genetic testing is so important, because it's really only through genetic testing you can identify what the genetic disease is and who has it. So in general, adult onset genetic disease is inherited in a dominant way. Now, some of you on the call, there are cardiomyopathies that are recessive, but most of the cardiomyopathies that are covered by by this consortium are actually dominantly inherited. And so what that means is that there's um, there's two copies of most genes in mom, two copies in dad, and if one of them is mutated, then that means that 50% of that person's offspring, 50% of that person's children are going to have that variant. And that's a coin toss for every individual one. You may see a family that has three kids and all three of them have it, and you may see another family where there's three kids and none of them have it. Um, so it's just a random 50-50 chance for each of the children. Um, However, because the risk of cardiomyopathy is not 100%, it sometimes looks like it can skip generations. Now, that doesn't mean that the gene is skipping generations. It just means that the disease is skipping generations, because not everyone who has the gene is going to get the disease. So why is family history and connection important? And the reason why here is because if you know about your genetic risk, you really have a superpower that can help other people understand about their genetic risk and potentially help them prevent disease or get an early diagnosis and find out about it and take steps to live longer and healthier. So, what incomplete penetrance means is that if you have a parent, this proband is going to inherit from a parent, and it may be that that parent isn't affected. So the parent didn't know about it, but maybe they didn't have disease. Maybe they died young from something else. And so they never identified that disease. And so that means that somebody might discover it for the first time without having an affected parent or grandparent. I think that this is going to advance. Okay. And so you can see that it can go down from generation to generation. Usually you can trace it up to a parent and sometimes a grandparent. Again, not everyone necessarily has it. And so here you can see if this is you, then each of your parents have a 50% chance of having it. Each of your grandparents have a 25% chance of having it. Your full aunts and uncles also have a 25% chance if they have the same two parents. First cousins are going to have a 12.5% chance. First cousins once removed, 6.25% chance. And you can see how the probabilities go 50-50 every time. So one of the things that Connect My Variant does is by connecting people, is help them find out which side of their family it came from. So if we can say we know which side of the family it came from, like we know that the grandparents for certain had it, then we can say, this aunt or uncle has a 50% chance, and now this cousin has a 25% chance of having it, and this first cousin was removed has a 12.5% chance of having it. And then there's a whole set of aunts, uncles, and cousins on the other side of the family that we don't have to worry about. So understanding which side of the family your variant came from can be really helpful. But if your parents and grandparents are deceased, then it can be hard to know which of them had the variant. So that's when it's really cool when you can identify other people who have your variant. Because if you can talk with them and find out if they're related to you or how they're related to you, then you can triangulate up and figure out which parent, grandparent, great grandparent potentially had the variant. And so again, if you can then identify up to which great grandparents, then you can go out to second cousins who have high enough of risk to getting genetic testing. I'm going to show a real life example here, and I have to have a caveat here that in Connect My Variant, we started working with hereditary cardiomyopathy about 2.5 years ago. But we don't just work with hereditary cardiomyopathy, we also work with hereditary cancer, hereditary hypercholesterolemia, and other hereditary diseases. We focus on the family outreach aspects, not on the disease aspects. And so this is an example from somebody who has a CDKN2A variant, and um, they are so the CDKN2A is associated with my melanoma and pancreatic cancer, and not cardiomyopathy. So that's the caveat for this. So this is Martha. Martha found out she had the CDKN2A variant. She had a family history of melanoma, and that's how she was able to identify that. And then there was also pancreatic cancer in her family. And so she was able to see which of her relatives had that and have a pretty good guess of which side of the family she thought it was on. Um, and then she made sure she had her kids tested. This is her son, graduating from college. She's getting a master's degree in bioinformatics at the same time. Her son has the variant, and he's now taking steps to be able to prevent melanoma and to monitor for pancreatic cancer. Um, so it can certainly benefit other relatives. And then she traced her variant through to her, well, she looked at the people who had melanoma and pancreatic cancer in her family and is pretty confident that it came from this is her mother, then her father, and then her maternal grandfather. So these are the family pictures that she found of the side of the family that she thinks it's on. And the interesting thing is that these relatives that she found, she's not exactly sure where it came from, but they all lived in the same area of Ohio. Um, and and so that's where she thinks she traced her variant to. As she went back down her family tree from her great grandparents, then she started looking for relatives who might also have the same risk. Um, and she lives in Arizona and near Phoenix, and she found some of her other family members who just by chance had also their parents had moved from Ohio into the Phoenix area. And so she was able to find these relatives who didn't know about the genetic disease in their family and set up a lunch appointment so she could go and went out to lunch with them and explained what she'd found in her family, explained her experience with this genetic variant, and asked them if they'd seen any of the same types of diseases in their family. And they were like, yeah, wow, actually, our parents had the exact same disease that you're talking about. And so I think second cousins are now getting genetic testing to see if they can do what they need to do to prevent disease. So this is another example. This is also another cancer family. This is a family that has a PALB2 variant. PALB2 is associated with breast cancer and ovarian cancer. And this one individual in this family uh, worked with Connect My Variant, posted on the Facebook group for the PALB2 Facebook group, which has several thousand people, asking people, does anybody have my variant? I want to see if I can figure out where it came from. And spend a lot of effort trying to connect these relatives. Ended up using ancestry DNA tests to do some triangulation, which the BOU Center Family History and Genealogy can help with. And so between these dozen individuals, they found one group that's in has ancestors in Virginia. This is the group that has ancestors in South Carolina. Another group that has ancestors in Virginia. And between the two groups, a common ancestor who died in 1688. Um, because of the efforts of this group of individuals, then hundreds of people have been able to receive genetic testing and can take preventative steps they wouldn't have been able to take earlier. So this whole pedigree this whole family tree took about 5 years to put together between getting everyone together, doing all the family history, and starting to do the outreach. And it's growing now. Since I put together this slide, then there's 4 or 5 other people who have found out about their variant, been able to tie themselves into this family tree. So one person's efforts bringing together other people and then working together can make an enormous impact in disease risk. Um, one of the things that's exciting about family trees that are this big, and one of the things that Connect My Variant has just started to do, we just got a research grant about 6 months ago to start for some of these more common cancer variants to start calculating variant-specific risk. Not every variant has exactly the same risk as the other variants, especially for missense variants or splice variants. And if you can get a big enough family tree, then you can calculate a robust variant-specific risk for that specific variant. Um, not the most of the families that we're working with are not quite big enough to be able to do that yet. But the goal in the long run is to be able to have everybody meet somebody else who has their variant, find out where their variant came from, and understand if their variant has the same risk as all the other variants in their gene, or if we can give them a better understanding of what the specific risk is for their specific variants. Oh, sorry. Yeah. The goal is that this is not going to be exceptional. This is an exceptional case now, but we hope that in the future this will be the normal thing that happens for everyone who has a pathogenic variant. They understand where it came from, who else needs to know about it, and what the specific risk is for that. So I know you're all asking now, how do you sign up? Here is a QR code that we made with Genetic Cardiomyopathy Awareness Consortium. So it takes us to this sign up page. And on the sign up page, you enter your name, email, phone number is optional. The key things are, do you have a mutation in your variant in your family? And then if you do, are you interested in sharing your contact information with others who have that same variant? So clicking yes here tells us that you want to meet other people. And when we share that information, we're sharing first name and email address. That's how we share contact information. And if you want to share other information, it's up to you whether you want to share other information to get to know each other better. So the other thing that's really important is that when you hear we're asking for your family's the gene and the variant. So I told you where to find your C dot or P dot on the page. Not every variant has a P dot, but they all have a C dot. And so find out on your report where that is, write the gene name and the C dot or P dots, and that'll help us know how we can connect you with other people. If you want, you can take a picture of your genetic test result and upload it, and we can find that information for you. Then there's a few other questions about what language you speak and where you came from, and if you're interested in research. So that's what's on the sign up form, and you can scan this QR code now and sign up now if you want. It'll be a few more slides as well if you don't get a chance now. I think it will go out after this presentation as well, a link where you can find this sign up page. Right now, let me talk about it later. So what can you expect when you sign up? So this is a quote from Michelle Springer. She has a pathogenic variant in a gene called ATM. She's a genetic counselor and has been doing genetic counseling for a long time. And she heard about Connect My Variant and thought it was really a great idea. And then about 3 or 4 months later, I ran into her again and she said, yeah, I kind of wondered what it's like for people. And I said, well, why don't you sign up yourself? Because you have a pathogenic variant in a gene that we help people connect with. She's a week later, I talked to her and she was just over the moon. She said, when I signed up, I got an email telling me about different resources I could find for the specific gene. Oftentimes, they're going to try like pointing people towards like the Facebook groups that the Genetic Cardiomyopathy Association Consortium is set up, where people can find other people with their same gene. If we know of a message board or a group with somebody for people with this exact same variant, we're going to provide that to you as well. That doesn't happen very often, but when it is, it's important for people to find that. And then she said within 2 days, I had an email from a real person asking if they wanted to, if I wanted help with family outreach, and if they could help me at all, they could connect up with a family outreach navigator. And then she set up that appointment, for a few weeks later. And it's the type of support for family outreach that I wish that everyone could have within the healthcare system. The genetic counselors are trained to do this type of work, but unfortunately, at least in the United States, genetic counselors aren't paid to do this type of work. They usually don't have an opportunity to help people with that family outreach. And so one of the reasons why we set up Connect My Variant was so that everyone could have that type of support. Now, right now, overall, only about 40% of people who sign up for Connect My Variant are able to immediately connect with somebody else who has their variant, which means that 60% we say, these are the resources we have, but we're not able to connect you up with somebody else who has their same variant right now. That for cardiomyopathy genes, it's a little bit higher than 60%. Is probably closer to 80% of people have to wait for somebody else to have to wait for some time to find somebody else with their variant. But that every week I see somebody else who has been waiting for 6 months or a year or 2 years, and somebody else with their variant signs up, and they get to be introduced to somebody else who has their variant and see what their experiences are and how they can connect. So this is our board of directors. You can find out more about Connect My Variant just by going to our website here. And there's a lot of volunteers. Every one of these people on this screen is volunteering to make Connect My Variant better. I volunteer my time for this, and we have lots of volunteer family outreach navigators and volunteer variant champions who help other people with their same variant. It's really a grassroots volunteer-driven organization. So there's the slide for how to sign up again, and I'm going to pause now and hand it off to Christy, and to your questions.
Great. Thank you so much, Brian. That was wonderful. And we do have some questions rolling in. I just wanted to remind you that if you do have a question for Brian that you'd like to ask for him to answer now, please go to the Q&A. So there's a chat and a Q&A. You want to go to the Q&A section and enter your question. So with that, thank you very much for having me. And let's go to our first question. So, Brian, can you please explain the difference between missense, nonsense, and truncating variants, and how do you find those on your genetic testing report? Oh, sorry, you are on mute, Brian.
If I can go, I wish I could go back to the slide. I don't know if we can go back to the slide that had those things. So a missense change is going to have one amino acid change to another amino acid. That's what a missense change. That's the definition of it. Then termination or a nonsense one is one that has a stop codon. So it's going to have amino acid, and then it's going to have the tear or a star by it saying kind of this is a termination codon. And then there's a frameshift variant, and the truncating variants could be either the ones with the tear on it, the terminating variants, or the nonsense variants, or the ones with the frameshift. Those are both considered to be truncating variants. So I hope that answers your question. They're technical terms. The more important question for most people is whether it's classified as pathogenic or benign, because there are some missense variants that can be pathogenic and there's some that can be benign. There can also be truncating variants that are very late in the protein that can be benign.
So are missense and nonsense the same thing?
Missense and nonsense are different.
Oh,
nonsense and termination proteins changes are the same thing. Um,
okay. Okay.
yeah,
Interesting. All
yeah.
right, good. And again, since we can't go back in the slides, this might be a little hard, but I have a question that is, can you help us walk through the protein again? There was some misunderstanding on what the P dot meant.
Sorry,
Yeah.
I'm just going to interrupt. Can you tell me do you know off the top of your head what slide it is? And I will pull it up if that's easier.
It's gonna be slide. Let's see. I don't have it. I don't have it at the top of my head here.
If you
Um,
give me a round and I'll pull it up. Give it a round. What slide?
Let me see
Even
here.
one of the genetic report slides,
Okay.
DSC or
I'll
lamin
keep
A,
on. If you
probably.
guys want to, here, I'm going to pull up right now and you can tell me if I'm
It's gonna
close.
be a, it's gonna be quite early on. So it's so probably about slide 20 ish. So so what happens is you have your DNA sequence, and then your DNA sequence is transcribed to RNA and then translated to protein. And so here you go. This is a good one. You can stop here. And so the C dot is going to give you what position of your DNA sequence has changed.
Yep.
And then the P dot is going to tell you what position of the protein has changed. And usually this number is these numbers divided by three more or less, because every three positions in the DNA codes for one amino acid or one piece of the protein. And so the P dot's going to tell you kind of what the change of the protein and what function that has, and the C dot tells you what's in the DNA. And every protein is different, and every protein is going to have for some genes missense. There's a lot of times that missense changes make a difference, and for some
Hmm.
genes missense, there's not very many missense changes that make a difference. Lamin A missense changes tend to make a difference. Titan, missense changes, most of them don't tend to make a difference. And so it's hard to tell, like you can't tell a whole lot from this protein change about what it does without knowing more about the gene.
Hmm.
But you are able to say kind of this is where in the gene it happens. And there's 20 different amino acids, and I'm not going to go through the whole kind of all the different amino acids and what they can mean, because that would be a freshman level
Medical seminar, yeah. I'll get my medical school
I hope that
doctorate.
answered the question.
Great.
Let's leave this up here because there may be other
Yep,
questions
let's leave
going
it
forward.
up. So the next question I have is from Carrie, and she's talking about having a pathogenic RBM20
Hmm.
truncating variant. She lists that. So Brian, if you
Yeah,
want to look at it, you
you can
can
see
see
it.
it in
Yeah.
the actual questions.
Hmm.
She says there's conflicting information on whether truncating RBM20 is more mild or not, especially as mine is outside of the typical hot spot zone. My
Yeah.
dad has it as well, is asymptomatic at 74. Two of her children have it. She's awaiting an MRI. However, because it's classified as pathogenic, her doctors are taking it very seriously. Where or who can I learn more from about my specific variant and what it means?
Yeah, that is a phenomenal question, Carrie. And I think that's the hardest question in medical genetics right now. So right now, as someone who is not your doctor, then I have to say you should talk to your doctors and listen to what your doctors say, because they're going to interpret your variant in the context of your family and the context of your medical history. And Connect My Variant, we have a disclaimer at the bottom of all our things saying we're not giving medical advice. So that's the textbook kind of information that I have to tell you. Now, one of the reasons why we started Connect My Variant is because your family tree is going to give you the best picture about what your variant means. And so knowing that your dad had it as an asymptomatic at 74 is really helpful. Like that shows that somebody can have this variant and live to age 74 being asymptomatic. If you knew that you had other aunts or uncles that had the variant and what they look like, that would give you other pieces of information about that. And as we try to connect people that have the same variant, one of the first things they do is start saying, what happened to you? What happened to your relatives? And it's the perfect thing for them to be saying, because what they're doing is collecting more information about what their variant looks like. Um, and we see people that see surprises, either because they see more than they expect, or because they see less disease than they expect in their family, or sometimes it's asymptomatic. A lot of people at older ages don't have any symptoms, and then all of a sudden somebody dies really young. So it takes a lot of data to be able to really understand if one variant is different than another. And part of that's because there's a lot of chance involved. Um, you know, Sometimes there can be a family that has a lot of disease just by chance, and another family could have not very much disease, also same variant just by chance. And so it takes about a dozen people with the variant to even start to get a good idea of what that risk looks like. And in the population databases, like I can say your RBM20 variant, they could sample a sample of a million different people and find just a few people with it, not enough to be able to give a good picture of what that variant is. But if you find somebody else with your variant, and if you really get lots of people in your family who have risk to get tested, then you're going to have more people in your family who have that variant, or in the families you're able to connect with who have that variant, than you'd see in any of the research studies. And so where or who could you ask who you can go to find out more about your specific variant, what it means? First, go to your doctors, talk to them. Then if you're not satisfied with their answers, the best place you can go is to your own family, and see what it means in your own family. Hope that answers your
Great.
question, Carrie.
Thank you, Brian. And then the next question is from Renee, and she's saying that she was diagnosed with ARVD in 1995. She had genetic testing in 2025, which showed she was positive for RYR2. Last year, she had genetic testing and nothing showed up. So why didn't the RYR2 gene show up on a recent test?
Yeah. So right now, it looks like you said that the most recent test was something called every gene, and I don't know too much about that. But it could be that the gene wasn't on that test, or it could be that they only tested certain variants in that test, and that this one wasn't on that. And so there are, like, if you go get testing through Ancestry or 23andMe and take your data out of that and plug it into a kind of software that's going to spit out medical stuff from that, usually that data doesn't have the same level of data that clinical sequencing would have. And so, I generally stay away from direct consumer genetic testing companies and the types of reports you can get from online things, because they're not going to have the same level of genetic information that a clinical test that a doctor orders or a genetic counselor orders is going to have. So without knowing specifically what the every gene test covered, then I wouldn't be able to say. But you want to make sure that the test you're ordering, or the test your family members are ordering, is targeted for the specific mutation in your family. Otherwise, they might. I've all the time I hear about people who say, "Oh yeah, I had a genome screen through some direct consumer testing thing, and it didn't have the variant that was in my family." And I want to say, no, 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 you didn't get the right test for that gene. You need to go to a doctor, genetic counselor, with a copy of the genetic test report from your relative, so they know what to test for, and so you get a result that's actually that's a confident negative, knowing that the test was able to detect that specific variant. I hope that answers
Right.
your question.
Thank you, Brian. Great. And the next question, just we are going to be sending a recording to everybody who's on this call and who signed up. So you will get a recording link after in the next few days. So, and we'll go over that more. Next question, I just as a comment, Jude, she says she's spoken to you, Brian, before, and this was very helpful. So thank you,
Oh,
Jude.
good.
So here's a good one from Stacy. So she's asking, how can three people in the same family have different variants?
That is a great question. So if you go back and think, if you actually, if go to the slide that's about two down from here, that has um, the two parents with the DNA things that's, let's see if we can do that one. And we'll see. As we're going through there, so each of us has 20,000 genes. And the cardiomyopathy genes, the ones that have been identified I think we're going the opposite direction where I'm going for. Um, but so each of us have lots of different genes that code for many, many different things. And each of them are inherited really independently from each other. And so when we talked about the slide they're going to, I talked about the coin toss to see, which children have the specific variant. Then, that's the one. Stop right there. So, if it's possible for um, mom to have two different variants in two different genes, and dad to have a variant in a different gene, and one child to inherit all three of those, and another child to inherit none of those three. Um, but I see all the time, you actually in the two genetic test reports that we flipped through, then both of them had two different things. And I look at them and say, oh yeah, one of them is the cardiomyopathy one that whoever has that result needs to worry about, and the other one is something that, because I know these genetic variants are ones that cause different risk for a different disease and maybe a different lower risk. So you should talk to your genetic counselor, talk to your doctor about what on your report is more important for you and what's not as important for you. Um, and actually, I can say that everyone has millions of variants. Most of them just don't get on the genetic test report. And so it's certainly possible for individuals to have multiple variants. And it's possible for people to have multiple pathogenic variants for the similar diseases as well. I've seen that happen multiple times. I hope that answered that question. So I
Thank
see
you.
I see the next question. So it's from Michelle. She says, I keep getting a message that something happened when you sign up. Michelle, when I was when I saw that, I actually looked, and it's because you signed up in March 2023. So congratulations, you're ahead of the curve. If the most common error that happens is because somebody's already signed up, and we use an email as the unique identifier. And so sometimes people click the sign up button and it works, and they're not sure if it worked, and then they enter the information again and they get an email saying something happened when you sign up. It's usually because you were already signed up. So thank you, Michelle. If any of you else
Right.
get that answer, feel free to just email info@connectmyvariant.org and say, I got an error message. I just want to double check to make sure I'm signed up. Almost all of the time, that's why that message
Great.
happens.
And maybe with the right variants too, right, Brian? What if someone's variants have changed or they have additional
Yeah,
ones to add?
they could
That would
they,
be a
they could
good way
just
to
send it.
email.
They could just send an email and yeah,
Okay,
great.
great. So yes, if you have additional information, go ahead and send them an email. All right,
Absolutely.
next question. Is there a way that I don't pass the mutation on to future generations? Is IVF a possibility of accomplishing this?
Yeah, that is a fantastic question, a very personal question. Um, IVF is available for people who can afford it and who want to take that option. Um, I know many families who have decided that they want to do IVF to prevent a specific mutation from getting passed down. I also know many families who've decided that it's not worth it postponing having their family members, and who have hope that or who have hope that the genetic changes that are going to be treatable enough in the future, and for many genetic conditions they are very treatable now, and so they've decided just to have children without thinking about that. But yes, for most specific identifiable variants you can identify, IVF can be a possibility. Um, Great
Thank you.
question.
Thank you, Brian. Next from Jude. My sister and I both have the same lamin A gene mutation, but also a novel missense variant on it. Does the novel missense interfere with how the laminin A mutation behaves?
That is a great question. Um, and that's something that depends on the specific mutation. I'd have to look at the specific variant to see what that means. Most novel variants that are classified as variants of uncertain significance tend to be benign. And I think it would be unusual for the two variants to interact with each other, but not impossible. So you really have to look at the specific variant.
Okay, thank you. Um, Next question. Anonymous is asking about the PowerPoint. I think we do the recording, but not the PowerPoint, but I will let Nikki clarify that at the end. So Nikki, if you could just clarify that when we're done, that would be great. Moving on to Jim. We now have 6 living relatives with the BAG3 exon 1 deletion after a cousin tested positive in 2023. Not all exhibit DCM. There are at least 5 deaths which appear to be related. In the 2023 report, it was labeled novel and pathogenic. Later reports say likely pathogenic. If it is in fact uncommon, what is the likelihood that we can learn from similar variants?
I'm not sure exactly what the question is. I know that if there are more people with the variants who talk to their doctors and who get reported, then it's more likely to be classified potentially as, not just likely pathogenic, but pathogenic. Those two are treated, likely pathogenic and pathogenic are treated similarly. Um, and so the medical treatment is going to be similar for all of that. Um, and if you're able to build out the pedigree to show that those 5 deaths are likely related to the BAG3 variant that's in your family, that could very easily help reclassify that variant.
So just can we take 1 second, Brian, and talk about the likely pathogenic versus
Yeah,
pathogenic? So what is that little difference between those two?
yeah. So that little difference is because when we don't want it, we don't need to be we don't want to force ourselves to be 100% confident that a variant is pathogenic before we start taking action,
Mhm.
because it's really difficult to get 100% certainty about anything anywhere. And so the variant classification guidelines say that you should be somewhere between 90 and 95% confident that a variant is pathogenic to start treating someone. And so at the 90%, we usually call it likely pathogenic. And then once we get to like the 95%, it gets changed to pathogenic. And the guidelines are used somewhat subjectively, so sometimes there's differences between laboratories. And because different laboratories have different amounts of information, you might see a variant that one laboratory classifies as likely pathogenic, another classifies as pathogenic, or one classifies as variant of uncertain significance, and another classifies as likely pathogenic, or one classifies as variant of uncertain significance, another classifies as likely benign. But the
Hmm.
the gene or the variant knows what it does, right? It's going to be the, it's going to it is what it is. Um,
Yeah.
but we don't know everything about variants, and this is a great example because it says that it was novel in 2023. So this may be a variant that's really only present in this family, and um, because of the characteristics of it, people were able to say that it's likely to be pathogenic, but because they haven't seen it very often, then they don't have the evidence to really say it's pathogenic. To be earlier, be over 95% confident that it's pathogenic. Did that help?
Okay, yeah, that definitely helps. And Greg, do you have something to add?
Yeah, it's interesting. I want to go back to the question before the RYR2 that didn't show up. I think this relates real quick. I mean, we work with Evergene and Vitae and Ambry to direct people to genetic testing, Brian, and Evergene's a CAP-CLIA certified, Broad Institute does the sequencing, MGB, LMM does the analysis. So the one thing I've learned is what's interesting. So my test that was up there, I think this is important for everyone. So when I was tested so my test was the laminin A 331 F2. That was me through Invitae. It was interesting that I did a few years ago. 14 years ago, I was tested through, I think it was GeneDx at Cleveland Clinic. And what showed up there was Lamin 331 as athenogenic, Lamin 399 as likely path, and TPM1 as a VUS. There's a couple things here. I think Brian hit on one is sort of the threshold of listing something as likely path, and path is not always exactly the same, so it show up in one test. A lot of the genetic testing companies may or may not report the VUSs because, the question is, what do you do with that? And Brian, please elaborate sort of on this more. And then and things are recategorized all the time. It's interesting. I mean, it's not an exact science. The fact that I know Layman 399 was a likely path, and they downgraded it to a VUS. So it's kind of over time. It's interesting how this all changes. It's a bit confusing. So I'll be quiet. Thank you.
Yes, thanks for bringing that up. I think that highlights another reason why you might see a variant reported on one test and not another. Usually likely pathogenic variants are not reported, and some laboratories don't report variants of uncertain significance. And so it could be that a variant is reported by one laboratory, seen by another laboratory, but not reported. And ideally, in the notes that the doctor or genetic counselor provides, it will say, this is the variant we're looking for. Please report about this variant. And in that case, some laboratories will comment on that. And if they don't, you can potentially ask them, hey, did you see this variant? And I've seen some patients who come to us and say, at Vanderbilt and say, where I do genetic testing, did you see this variant? And we're able to go back and say, oh yeah, we saw this variant. We classify it as likely benign. That's why it's not on the report.
Yeah.
So that's another reason.
But if you have the disease, you have it. But again, it's just the science is getting better, but it's not exact yet. If you have disease in the family, you have genetic cardiomyopathy. There's a lot of people also VUSs. Do you report, not report? Does it make you sick? Not, we don't know. Then there's also a thing called gene elusive, where there's a lot of people that have probably some type of inherited cardiomyopathy, but they just don't know the gene yet. So unfortunately, it's not an exact science.
And there is familial disease that's not genetic, that a few people in the family have the disease, but there really isn't a clear genetic. There's two other questions here I want to answer before we run out of time.
Okay.
One of them is, if someone has the exact same variant as you, does that mean they are definitely related to you're definitely related to each other? And the answer there is no. But the analysis we've done suggests that across these inherited diseases, there's a 90% chance that if someone else has the exact same variant, you share that one. Now, there's some mutational hotspots where it's less likely. It's more likely that those variants kind of occur by chance. I know that there's one Lamin A group where we got 5 people together who had the same variant, and it seemed like, and 4 of them said it was de novo, meaning they were the first person in the family who had it. So that specific Lamin A variant, absolutely not. It doesn't mean they're related to each other. There are some variants that are known as founder variants, where if 2 people have ancestors from the same region, it's much more than 90% chance they inherit that from a common ancestor. So if someone has the exact same variant, you're not definitely related, but there's a good chance you are. So I wanted to make sure that that's clear. So the 2 reasons why we introduce people, one is to potentially find common ancestors. The other is because there's a lot of information that people can find just from talking to each other, even if they're not related. The other question I see that's really important, an anonymous person saying, they have a 23-year-old son was diagnosed with heart failure. They found a variant, and their husband tested positive for the same variant. But it says if the variant's not detected in his 3 brothers, is there any chance that his brother's 3 children could have it? I.e., can it skip a generation? And the answer there is no. If the 3 brothers got testing for that specific variant and it was negative, then there's no chance that the genes can skip generations. So genetics doesn't skip generations. Now, if the brothers haven't been tested, the brothers don't have the disease, but they haven't had genetic testing. So let's say the brothers don't have cardiomyopathy. It could still be they have the gene, but haven't developed cardiomyopathy, because not everyone who has the genetic variant gets disease. We heard about somebody earlier who had a father who's 74 years old without having any heart disease, but had the pathogenic variant. And so the what we call the phenotype, the cardiomyopathy can potentially skip generations, but the gene doesn't skip generations. And so the genetic variant doesn't skip generations. So if the brothers tested negative for this specific genetic variant, their kids won't have it. And that's actually one of the great benefits of genetic testing and that can help people get genetic testing is to tell them, well, if you test negative, your kids don't have to worry about it at all. So those are the two questions are really common questions I wanted to answer. I know we're at time.
Yep.
I don't know if we have time to keep on
Yeah,
doing
Brian, do we have
questions.
to, or do we have time to go a little bit further? There's a couple others that I think would be important for the whole group to answer. Do we have a few more minutes? Brian?
I have a few more minutes. I
Okay.
didn't know if you were
Okay.
asking Nikki if we have a few more minutes.
Okay, I think Nikki said 50, we could go up to that, and I know she still has some,
Okay.
some work. So the
Great.
question I think is important is, I have 3 variants, one being pathogenic. How often should a person be testing?
So if somebody knows about their genetic test results, then they shouldn't need to get genetic testing again.
But what if they're unknown? So only one, she has 3 variants, one is pathogenic, so those other ones may be uncertain, for example.
Yeah.
Should she be
That's a
testing
good
more?
question. I would say that you don't need to get genetic testing again for that.
Okay,
But you can talk to the genetic counselor or doctor who gave you that result and ask them if there's more information about that variant. So you
okay,
shouldn't need to get genetic testing again to get a reinterpretation of those variants. And you should be able to ask them if they have any more information about those variants. So I would say don't get genetic testing again. If you've lost contact with the doctor who initially did the genetic testing, then you can ask your doctor again, or ask to get an appointment with a genetic counselor and ask them if they can look up those variants and see if the interpretations have changed.
right. Now, if you didn't find the variant, but still have family history, maybe genetic testing should be done again, right? Because they may find new variants that have come up in the last 5 years or
Yeah,
something.
that's true. And there have been major shifts in genetic testing.
Mhm.
I know that when we were doing genetic testing a decade ago, we just did targeted genes. And so somebody might not have had all of the genes that have been identified. If someone's done genetic testing in the last 5 years, it's unlikely that there's a new gene or a new cause of cardiomyopathy or that they could potentially find. And so I would say if someone's got genetic testing in the last 5 years, it probably doesn't make sense to get genetic testing. There is new technology that's coming out now for like long read sequencing and that has an ability to detect different types of variants that we haven't been able to see before. Not really clinical prime time yet. And so if I had genetic testing 2, 3, 5 years ago, and tested negative and had a very strong family history, I'd wait another 4 or 5 years till that technology is common before I got genetic testing.
Okay, thank you, Brian.
Yeah.
Here's one that just talks about your patient advocates. This is a question about what they do. Could
Yeah.
patient advocate assist with dealing with insurance company who don't classify rare disease as a disability?
Yeah. No, we can't help
No.
with that. Sorry.
Yep.
These are family outreach navigators, and they're volunteers, and they're really trained in how to help people talk to their family members, and that's all they're trained in. So we don't typically help with healthcare. We help with family outreach. That's a good question, though.
Okay. Um, and so this one was I was tested in 2015. Should I be tested again? I think we answered that. And this one, what is the earliest age for a genetic mutation to show up on a testing? A young family member wants to know, but the doc said they are too young.
Oh yeah. So when they're saying they're too young, that's a really good question. So your genes aren't going to change over time.
Right.
You're going to have the same genetic variants, but the American College of Medical Genetics has recommended that for adult onset diseases, so for diseases that are unlikely to affect somebody when they're a child, then they've recommended not to get genetic testing until someone's an adult. Now, if there's a gene that starts showing up, if the effects start showing up in teenagers, then that person might want to get genetic testing when they're a teenager. But the doctor might say, well, this person's a 2-year-old, it doesn't make sense to get genetic testing if we know they're not going to have any symptoms until they're 18 or 20 or 30. And so that's a general principle is not to do genetic testing until there's action we can take. And so that may be what the doctors are saying about the person being too young to get genetic testing. I don't know what the gene is, and I don't know what how early the effects of that happen. But I would say that's probably the reason why they're saying don't get genetic testing, or
Okay.
to wait to get genetic testing. And the principle behind that is to give children a free childhood, as worry-free childhood as they can have, and to wait till someone's old enough to be able to make informed decisions about their own health care, as opposed to having someone else make the decisions about their health care.
Okay, thank you. Nikki, can I ask one more?
Sure, one more, Christy.
Okay, one more. That's it. Because this is kind of an interesting one too, I think. My result says negative, but on the report it states there is a variant, and she lists a variant which is in GLA. What does that mean? If there is a variant, but it's a negative result.
That is really interesting. I haven't looked up the variant, so I'm not sure.
Yep.
If the variant's classified as there's a few reasons. If it's classified as a benign variant or variant of uncertain significance, there may be some reasons why they want to report it. I know that on our test results at Vanderbilt, we have kind of two lists of variants, one that's the variants that are important and one that's kind of an additional list of variants that we want to say that we saw just in case somebody comes back and asks, or in case they come up in the future, but they're not. They could be carrier for something that's recessive, so it could be a variant that you need to have two copies of the pathogenic variant in the gene, and so if there's only one, then it's still a negative result because the person just is a carrier for that. So there's lots of reasons. There's a few different reasons why someone might have a negative report. Uniformly, that specific variant shouldn't influence medical treatment, unless there was a mistake on the reports. Then usually, then those variants shouldn't influence medical treatment. But the reporting laboratory wanted to make sure that they let the doctor know they saw it.
Okay.
And oftentimes that's just because somebody gets genetic testing at another laboratory and it comes up again and they say, well, did you miss this? And I say, no, we didn't miss it. We saw it. We just don't think it's important for medical care.
Okay, got it. Interesting. Thank you so much, Brian.
Although I haven't looked up this variant, so don't quote me on this one not being important for medical care.
Absolutely. We will not quote you on that, and neither can you, Jennifer. So anyway, for the rest of the questions, I'm so sorry we didn't get to them all. I would have loved to do that, but unfortunately, we run out of time. So I'm going to turn it back over to Nikki. Thank you very much, Nikki.
All right. Thank you, Christy. And thank you again, Brian. That was fascinating. I know we all learned so much. I do have just a few final notes to wrap up. So thank you all for staying with us here. The Genetic Cardiomyopathy Awareness Consortium is helping connect patients with several genetic testing companies that offer testing to cardiomyopathy patients and their family members. So you can scan the code or visit our website for testing options. And next slide. As stated earlier, registrants will receive an email with a recording of tonight's webinar. We don't send out the PowerPoint slides, but you can see the slides again and again on the recording and watch it at your own pace. So the email will include a link to the genetic testing options, as well as a link to sign up for Connect My Variant. So if you weren't able to take any notes tonight, you'll get all of that in an email. I also want to state, I want to remind you to register for our next webinar, which will be held on March 11th, where we'll discuss genetic dilated cardiomyopathy, insights, research, and 2026 clinical trials. So registration is open and information can be found on the DCM Foundation and GCAC websites. And remember, even if you can't attend live, go ahead and register and you'll receive a recording via email. And we did share the QR code for Connect My Variant, but I want to thank all of our sponsors whose logos you see on the screen. Their support allows us to present this very important information. And thank you again to all of the patient partner groups for their ongoing support. We have to thank you, Justine, for your technical Zoom assistance behind the scenes. And most of all, thank you all for joining us tonight and for being such an engaged part of this important conversation. We hope you're leaving with clearer answers, greater confidence in understanding your variant, and a stronger sense of connection to a community that's here to learn and move forward together. So be well, and we'll see you very soon.